Well, thank you all. It's an honor to be here. I'm with the Nature Conservancy. Everyone calls it the Nature Conservatory, but we are the Nature Conservancy. I've been in Tennessee for 11 years, started work doing biological inventories, and that was really a break-in for me. I hadn't done bat work or bug work underground. Prior to that, I was just a general biologist and ecologist, and I really fell in love with it. And by dumb luck, was able to study under, one of a small handful of cave biology specialists, and just kind of got lucky. Wanted to set the stage a little bit. So this is a generalized karst map of the world. I can't remember, Hank, it might have been that had the definition of karst and mentioned, or maybe Patrick, that there is some argument about it. But my understanding, and the story I've always been told and sticking with, is that it's a German word for the Karst region of Slovenia, where sort of that landscape was first defined. And you can see there's plenty of karst in Europe and in Asia as well. And North America, it's not too shabby. Here in the U.S., some notables. Of course, I'm a biologist, so I always kind of pick out the biota, but Edwards Aquifer in Texas is just tremendous and has just incredible ecological aspects and resources. The Ozarks, Sid mentioned those big springs. If you've got some interest in karst hydrology, it is worth a trip to the current river system and Jacks Fork River systems to go see those huge springs. They just boil up from out of the ground, and it's unbelievable. I have no idea the flow rates. I'm not a hydrogeologist, but they're really something to see. Of course, Florida aquifers, and here we are in Tennessee, and you can see lots of karst here. Talking about it all day. You guys know we've got it. Notable is that we have more caves than any other state in the nation. We have over 10,000 caves now known from Tennessee. We add more every year. We lose a few too to technology, since this Tennessee Cave Survey volunteer group that's been talked about a little bit today. Since GPS and GIS has become available, we've discovered that sometimes caves were turned in more than once, sometimes 3 or 4 times, but when people plotted them on a map, they plotted them in slightly different locations. So we lose a few caves every year as people go out and geo-reference those sites and verify them, but people are finding new caves. So about 20% of the caves known to occur in the United States are in the state of Tennessee. And our standards for the definition of a cave are higher than most other states. Our standard is, of the Tennessee Cave Survey, 50 feet long, and it's either 30 or 40 feet deep if it's a pit. I can't remember which it is. But other states like Kentucky, they measure, I think it's 3 body lengths. If you get 3 body lengths from a drip line, you can call it a cave. So it's very significant cave resources. And it doesn't occur everywhere. It is unique. And you guys know that means water moves underground, but you just have this whole habitat and landscape with underground spaces, and it doesn't occur everywhere. And it's sort of a bonus for biotic organisms. It's a whole landscape that doesn't occur everywhere that they can occupy. So you get, aquatic organisms. You get what we call troglobites or subterrestrial organisms, things like bats. You have these habitats that are really unique and interesting, and the animals that occur there are just as interesting. This is just a pretty basic primer of cave biota. I'm probably not going to get as in-depth as Sid would like, but I understand we may have a little extra time, so we might get there. So my area of focus is anything that lives underground, and that covers a wide range. So there are things like deep soil organisms, edaphobites, things of that nature that are pretty hard to study. They're not necessarily karst related, and I tend to ignore them and pretend they're not there. So we've got this epikarst zone, and that was mentioned earlier. I think somebody spoke about epikarst a little bit earlier. We've got these different types of cave organisms, and we'll go through these. For my planning and conservation work, we also talk a lot about other things that are related to caves and karst, things like spring endemics. An example would be like the royal snail in Jasper, Tennessee. Known to occur, and I think that snail is only one site or maybe two sites, only known from these karst springs. And then what we consider limestone lovers, and they're organisms that either require some amount of calcium carbonate or they're very closely linked with limestone or calcium carbonate landscapes. So that could be plants on cedar glades, mollusks, things like snails and mussels that require uptake of calcium carbonate to survive. We're not going to talk about these today because they're not necessarily cave related, and we'll stick to these more cave and karst deals. So epikarst is, 1973. It's still a relatively new concept. I think Mangan, this was the first definition of karst, was 73. And it's just sort of that layer at the top of the bedrock where, sort of the soil meets the karst, and you have this zone, and it's often will be a water storage area, but certainly creates a lot of interstitial zone for organisms to live. It's a lot of ostracods, copepods, things of that nature. Typically pretty small stuff. And these are copepods here. And these systems tend to be carbon limited. So the dissolved organic carbon, rather than nutrients, is what seems to limit these organisms' ability to live and thrive. In other parts of the world, they've studied epikarst a lot. Here in the United States, we've not, as far as biota goes. Places like Slovenia, they have conferences on epikarstic organisms that draw hundreds and thousands of people, sometimes, if it's a big one. They have an international congress of epikarstic biotic organisms, and when they have their big event, it's a lot of people, but it's just something that hasn't caught on that much in the United States because these areas are so hard to get to. You can do Beau Rouge pumping, but really the main way to find these organisms and learn about them is to set sampling buckets in caves under drifts, and you're just getting things as they're dripping through from the epikarst coming into the cave environment. So we'll sort of move from the least adapted to the most adapted cave organisms. And there are also accidentals when you wander into a cave. If we stayed very long, we would be considered an accidental. You'll find lots of things that may not necessarily want to be in a cave. They may have fallen into a sinkhole somewhere or just wandered in because it was a cool place. And once they get into the dark zone, they bite the dust and become food for everything else. But beyond them, you have some level of adaptation. So troglo and stigo. Troglo is sort of subterrestrial. Stigo is subaquatic. And zine means user. So these are animals that use caves for some part of their life cycle. Typically, it's either hibernation or maybe it's just a nice, cool place to hang out during a hot summer day. But these animals are really, really critical to cave ecology. They're not necessarily cave adapted. They're not usually dependent on caves, but they bring in material. This wood rat, for example, wood rats occur in thousands of caves across the state, and they bring in material from outside into the cave, and that becomes food for the rest of the cave. Bats, same thing. They're foraging outside of caves and bringing in, the energy source into the cave. Crickets do the same thing. And some of these troglozines, even though they're not necessarily cave adapted, and they can't complete their life cycle in a cave, they're dependent on caves. We have, here in the southeast, we have gray bats. They're an endangered species, and they use caves both in the summer for their maternity colonies, their bachelor colonies, and in the winter for hibernation as well. And that's the only place they occur is in caves. Every now and then we'll find them under a bridge or some man-made structure, but almost never using any other type of natural habitat other than caves. And in the wintertime, they can be in hundreds of thousands, and they're pretty darn hard to count. We talked a little bit about LiDAR technology. Mammoth Cave has a project that I hope they're going to go through with, next year at one of their gray bat sites, using LiDAR to measure the volumetric difference in the cave when the bats are absent versus when they're present, and taking volume of the bat and trying to extrapolate, how much volume, how many bats were in there. Because now we go into these caves with 500,000 bats and do a very rough density in square footage, and we sort of track trends over time, but we really have no idea how many bats are in there. In the summertime, these bats are much more sensitive, especially in fraternity colonies where they form larger, typically 5 to 20,000 bats or so. We try to not go in there. Well, even if we did go in there and try to count, we couldn't really do anything because they're flying all over the place. But we do emergence surveys. But there are, thousands of caves across the landscape, and it would be impossible for us to go to each one of those sites and try and count. This is an example of a guano pile from gray bats, and that is here in Tennessee. So it's just an example of the amount of energy that they're bringing into these cave systems. So troglophiles and stigophiles, these are the more cave adapted. These are, the phile means lover. So these are animals that can complete their life cycle in a cave. They're frequently found in caves, although they may also be found just as happily in your basement, under a rock somewhere in the woods, some other cool, dark, damp place. Got some ecology in action. This was actually this Tuesday. I caught this guy's cave salamander eating a Pseudophilus cricket. That was a good find. So crickets going outside the cave, feeding on detritus, leaves, things of that nature, bringing that energy back and getting gobbled up. So this is amphipod, very common cave organism. Isopods and amphipods are probably our two most common aquatic organisms that you'll run across in streams if you're looking for them. They tend to be pretty small, and you have to really look for them. This is a glowworm. We call them glowworms. They're not really worms. They're maggots. They're a fungus gnat larvae, but they are bioluminescent, and we have them here in Tennessee, some of our caves, in pretty good numbers, and they can put on a pretty good show. And they're kind of one of my favorites. They are larvae that spins a web, and they're predatory, and they sort of dissolve and absorb their prey. They're pretty unique in the world of cave organisms. But for cave biologists, it's this truly cave-adapted organisms that we really seek out. These are the troglodytes, stigobites. All of these animals can be found nowhere else on Earth. They only occur in caves. There's some argument about how that came to be, but generally in this part of the world, people think that during the last ice age, people or these organisms were sort of looking for habitat that wasn't freezing, and they got pushed underground and stayed there. So I've got a couple slides of these guys, and all of these photos were taken in Tennessee by one of my colleagues with the Nature Conservancy that actually works over in the Ozarks. So this is an aquatic isopod, and very commonly found in cave streams if you look for them. Springtail, which springtails are a whole group of organisms that inhabit just about every habitat on Earth. And the cave-adapted ones are really interesting to read. Of course, the spider there. So we've got pseudoscorpion, dipleuron, cave snail, and this little guy is fairly famous. It's a pseudophthalmus beetle. It's a cave beetle. Something unique about Tennessee is we have all of these caves, all of this available habitat, and a really unique suite of organisms that occupy that habitat, but relatively no federal protection status for those organisms. These animals have really small ranges. They're inherently vulnerable because of their inability to move around from cave to cave because they're isolated in these small systems and they're kind of stuck there. For whatever reason, we don't have any that are listed. The pseudophthalmus beetles, there are 7 species that are candidate species for the federal endangered species list. They're under review right now. If they were to become listed, I think it would change the conservation landscape in regards to cave biota a little bit in Tennessee, and probably enable people to do a lot more surveys on these organisms and learn a lot more about our cave animals. We don't know that much. I'll dig in a little deeper here in a minute, but I'll say we don't know a lot, just because of the remote nature of these animals, their size, and their metabolisms are really low, so we can't do long-term studies on population effects. But we do have this really unique suite of organisms underground. So when we're talking today about all of these water quality issues, it's not just water moving through these systems from an upland to a major river and how that's going to affect the river system. There are a lot of things in between that are to be taken in consideration. And water is very important for these animals. So some of the bigger troglobites and stigobites that people are more aware of, they're more evident. Even the casual spelunker walking into a cave is going to notice these guys. This is a Tennessee cave salamander. We've got our two vertebrates here, Tennessee cave salamander and southern cavefish. And then, of course, the cave crayfish. So some differences between these animals and their surface relatives is, this level of adaptation to the cave environment. You see they tend to be eyeless, colorless, or I should say they produce no pigment. A lot of these animals tend to have sort of a reddish hue, but they don't produce pigment. That's just sort of the natural color of the chitin that they produce. Cave crayfish, just an example of metabolic rates. All these animals are sort of trapped in this cold, damp, low nutrient environment. Cave crayfish have been recorded over 70 years old. The surface relatives, 5 years is pretty well the max for a surface crayfish. This is a scan of an old Time Life book, and I'm probably breaking some copyright, but it's a really good, I had this book when I was a kid, and it's a really good example of differences between cave adapted organisms and their surface relatives. And so, they have no need for eyes, so their visual center receptor in the brain is much smaller, and that makes space for other things within the same, cranial capacity. So they have enlarged centers for things like smell, spatial memory, things of that nature, so they sort of make up for the senses that they don't have and that aren't available to them because of the darkness. If you've ever watched a cave fish swim, they're very smooth, agile. They don't expend energy that they don't have to. They just tend to be pretty slow and smooth. And that's just an example of reduced metabolism. They don't waste any energy. They're very graceful, and they breed very infrequently. Some of these organisms only breed once every few years. Just again because of this nutrient poor environment. So talk a little bit about sort of troglazines and their ability to bring energy into these systems. This is just a basic, really old food guide pyramid for the cave system. So you can think about a cave. Obviously there's no sunlight, and if there's no sunlight, there's no plant life. And that's what kicks most things off on our planet, is sunlight and plant life. But without that, these systems are completely dependent on outside sources of energy. There are some mineral-based contrasts to that, but they're pretty rare, and we don't have them, at least that we know of, a lot here in the U.S. But generally, energy has to come from outside, and it's organic debris either through a sinking stream, something of that nature. But the most consistent and the most usable source of energy for these systems is just really basic dissolved organic carbon that trickles in from the epicars. And the reason it's the most important is because it's so consistent. Generally, because the epicars function as a storage capacity for this water and this dissolved organic carbon, it trickles that down all year. And, despite what's going on in the landscape and our seasonal rain events, So it's just slowly trickling this dissolved organic carbon in, and then we get these seasonal flushes, typically of detritus and deciduous leaves and things of that nature. And most of our cave organisms, that is when they breed. They breed in the fall, at least here in this part of the world, when this deciduous matter is coming into these systems. All right, so biogeographic patterns. These animals are pretty isolated. The aquatic animals a little less so. They have a little higher connectivity, systemic connectivity than the subterrestrials, the troglodytes. But still, they have relatively small ranges and tend to be confined within a physiographic zone, sometimes even within a subzone. So this is some of our more common organisms and their ranges. And you'll notice, especially in East Tennessee, so somebody earlier today had really good geologic slides, three-dimensional, and showed these nice horizontal bedding planes in Middle Tennessee and the Cumberland Plateau. And that does allow really high levels of hydraulic connectivity, so that these organisms can move around a lot more, and their genes can flow a bit more freely in a much larger area. As soon as you get further east, where the limestone and everything is turned up on its side, you get little pockets of karst. And if you have little pockets of karst that's not connected, these organisms aren't able to get around as well either. So you have much higher levels of endemism. Organisms that are species known from one site or one county. We have much higher levels of that in East Tennessee than we do in Middle Tennessee. So those were two of the stigobites. This is a troglodytic beetle, and you can see these guys just generally have smaller ranges. This is just one example of biogeographic range of the troglobite, but that's pretty well the norm. They tend to have smaller ranges. Just because they're a little more isolated than the aquatics. All right, so what we know about cave biota in general, and it's not just here in Tennessee, but it's very little, because you have to put in a lot of effort to get a little bit of data on these guys. So the top map is caves in the state. Bottom map are caves that we have some biologic record from. And that can be, a grad student went in there in 1954 and, noted that there was a bat in there, or it could be a full-blown biologic inventory. But most of these are single record sites. I think for Tennessee, we've got probably less than 400 caves that have had full biological inventories done out of 10,000. So we really don't know that much. We have a lot to learn. But this is from a really good paper that David Culver put out in 2003. David Culver is a cave biologist at American University, and was really the first guy to kind of try and put a lot of data together and pull a lot of cave data together for large areas, not just here in the United States, but also in Europe, and try and model that information and figure out what it can tell us. And one of the things that he knew pretty early on, and I was slow to figure out, was that you don't have to do a full biological inventory in every cave within your political boundary or area of interest to do some basic planning and understand the concepts of what's going on underground. All these cave and karst systems, they have really high reported rates of endemism, but they're ecologically uniform. And that's a concept that as a conservation planner is very important. We know we don't know every species that's out there, but we know the ecological systems well. We know the basic suite of organisms that are going to occupy those niches and how they operate and what they need to survive. We know their key ecological attributes, threats, and all of those factors without having to know the individual species. And he just points out that they are inherently vulnerable. It's just the nature of being isolated in these caves, these small environments where they can't really get out and move around. They have these small ranges, so they're just inherently vulnerable by nature. And Tennessee, I mean, obviously, if you have more caves than any other state, you're probably going to have more cave biota and diversity than any other state as well, and we do. Despite being studied probably a lot less than states like Kentucky, Virginia, and Texas, the little bit we do know about Tennessee really shines. So troglobites, again, those subterrestrial organisms, were the top of the list. Cigobites were down here, we're tied for third with Missouri. And this data's a few years old. Matt Niemiller was mentioned earlier. He's been doing some really good work continuing biological inventories in Tennessee. And I would bet that we are probably number 1 or 2 right now, because he's been hitting it pretty hard for the last 2 years. But cave obligates in general, we're tied with Texas, which is really saying something. I mean, Texas has really significant karst resources, you know, around the Edwards Aquifer and beyond. But because they have federally listed cave invertebrates, there's been a lot of survey work done there, much more than in Tennessee. All right, so that's most of my talk is just about the organisms that live underground. I don't know how much time I have to talk about sustainability.
About 8 minutes.
8 minutes, okay. All right, fair enough. So we'll touch on it a little bit. So we know a lot about these organisms that live underground. We know a lot about sort of their key ecological factors, what they need to live. I've been involved in the last 1.5 years with a project that's funded through the Appalachian LCC. And it's basically looking at cave fauna in the Apps and the interior low plateau where we are now. And trying to develop, our initial goal was to try and develop sort of a decision-making tool for projects. And Fish and Wildlife Service right now is really big in taking all the information you can, rolling it together, and coming up with these decision-making tools. And it's a pretty complicated process, but what we did is we took all of the known records and put everything in one place. So, including bats. So it was bats, it was bugs, epikarst organisms, everything we could find a record on that was a cave endemic. We put it all in one place and then applied different environmental variables. We hired a modeler, Mary Chrisman in Florida, to take all hundreds of different environmental variables, overlay, and see which ones fell out as being more predictive in regards to where these animals occur. And the most important variables seem to be soil characteristics and biomass. And what we think is that that's just another indicator of that dissolved organic carbon and the soil's ability to retain water for a long period of time. It slows it down, it's able to retain it and move water slowly into the epikarst and just have a much higher retention time, so it has time to feed that cave system on an annual basis, and also to pick up carbon. It has residence time in the soil, so it's picking up carbon from the soil or detritus above it. And then outside the ILP, in the Appalachian proper, percent karst was really important, and it seems like a no-brainer, but it didn't fall out for the ILP. And we think the reason it didn't is because there's so much karst. I mean, if you look at the interior low plateau, it's probably 75% karst. And we think that sort of washed out in the data because it's sort of everywhere. But what was important for the ILP, and I didn't put it on here, was mean-based flow. And that's something you all in this room probably know a lot more about than I do. But that was an important variable for most of our organisms. And we did this organism by organism, sort of by suite, and we typically would go by genera. And we did everything individually, and the model worked pretty well on everything except top predators. And I think for a few reasons. One One of which is we have a lot less records for top predators because they tend to move faster and people don't see them as well, and they don't come to pitfall traps, so we don't catch them when we're out baiting and trapping in these cave systems for invertebrates. So our big suite of threats to these systems are going to be anything that's going to alter, the timing of water moving through that system and the ability of dissolved organic carbon or nutrient input through those seasonal streams and things of that nature. Altered hydrology, water pollution is an obvious one. And we tend to focus on these areas of surface and subsurface interactions. So that's anywhere there's a cave entrance or a sinkhole or a sinking stream, something of that nature. Those are really your most important places. Cave entrances are an obvious one. You've got animals coming out to feed. You've got crickets, you've got wood rats, things like that that are living in those caves, coming out to feed and bringing that energy back in addition to the hydrologic resources. All right, I would be remiss if I didn't at least mention white nose syndrome. So I've been working on this for several years now, and it has sort of taken over my life a little bit. Really sad story of the world of commerce and travel that we live in. You can be anywhere in the world in 48 hours, soaking the bacteria and fungus on your shoe that you're carrying with you, and we think that's exactly what happened. So this is a fungus that affects cave hibernating bats. It is identical, it's the same fungus that occurs in Europe and Asia. And presumably somebody probably just, had it on their shoe or their boot and was in a cave in Europe or Asia and brought it over to New York. And it's kind of a typical epidemic spread. It started in a few counties around there and then expanded out in 2009, made a big jump down the Appalachians and north into Canada. And it's really exploding. And it's having drastic effects on our bats. I think very conservative estimate is that it's killed about 5.7 million bats at this point. It's a fungus. It disrupts their hibernation cycle. Our bats tend to be, they're very small. They're from 4 grams to maybe 11 grams. And they have about 2 grams of fat to get them through the winter. And our temperate winters, it's cold, there's no food availability. All of our bats are insectivorous. So their strategy for many of our bats is to hibernate in caves. And it's a relatively safe, undisturbed, dark, quiet place, and they can bring their body temperatures down low enough to reduce their metabolism so that they can survive on 2 grams of fat to get them through the winter. And the high humidity really helps with the regulation of water balance. But this fungus disrupts their hibernation cycle, causes physical effects where it causes necrosis of their wing tissue, but probably more importantly, it just causes them to wake up about every, as much as every 48 hours. Bats are waking up because they're disturbed and they're trying to groom this off of their bodies, and they just use up too much energy and don't make it through the winter. In Tennessee, we have 16 species of bats. About half of those are cave hibernators. Of those, right now, only 4 are really getting hammered by white nose syndrome. Ironically, we have 2 endangered species in Tennessee that are bats, Indiana bats and gray bats. Gray bats so far, I wouldn't say they're unaffected. We're seeing behavioral changes, but they're not dying. So far, we still have a couple million bats in the state. Our numbers haven't drastically changed as far as we can tell on gray bats. Indiana bats, we only really had one really significant site, and that was in Cades Cove. And that site has been wiped out. Our smaller Indiana bat sites seem to be hanging on. We're seeing no drastic effects. But our more common bat, like this tricolor bat, used to be called pipistrelle, thank you. But the taxonomists never like to leave anything alone very long. But it used to be one of the most common bats you would see. You could walk in any cave in the state and probably see, a number of them scattered as you were going through, and they're getting hit really hard, as well as northern long-eared bats and little brown bats. That's all I get. Any questions? Do I have time for questions? Any questions?
Any questions?
When you do your studies, because you do have the diminishing populations, do you do anything to decontaminate yourself? I mean, do you have special equipment you wear into the caves?
Yeah, so I have regional equipment, and there are very strict decontamination protocols, and you can find those on whitenosesyndrome.org.
Now, I mean, I went to Mammoth Cave in high school, so 20 years ago, and, you know, it's just a tourist attraction. You can go in and out. Do they do anything now to?
Mammoth Cave actually does. Most commercial caves do not. Mammoth Cave does. They have a mat that you walk across. It used to be quaternary ammonia compound. It was discovered a couple of years ago that that's an off-label use, and they are a federal facility, so that was nixed, and they're now using a woolite solution just to physically remove any material that's on your shoes. But you do at least have to walk across this mat, both on your way in and your way out. Yeah. And the decontamination protocols, I should point out, it's good not just for white nose syndrome, but it's, you know, to hopefully for what could be the next white nose syndrome. I mean, cavers, I think a lot of cavers are pretty savvy about microbiology, and they understand the ecosystem more than the general public. You know, cavers that were exploring Lechuguilla 20 years ago were following very strict decontamination protocols, very similar to what we do for white nose syndrome now. It just took a bit too long to catch on, I think. Yeah. Any other questions? Yeah.
If they survive the winter with that syndrome, does it go away or do they have it for the rest of their life?
Yeah, they recover quickly. So the fungus is a cold-loving fungus, so it thrives really well in our caves where it's cold and dark and high humidity. But as soon as those bats warm up, that fungus starts to die. They can clear it really pretty quickly. If they've had a lot of physical damage, it could be a couple weeks, but they can come out of it. The problem is that the caves serve as reservoirs for the fungus. So as soon as they come back to hibernate, they're sort of in the same situation. The cycle starts over. There is a lot of research. It's an emerging pathogen, so we don't know a lot about it. We're still learning, but there is some really good research going on in regards to controls. And I'm really hopeful within a couple of years we'll have some tools to combat the fungus. Questions about cave biota?